Good. So welcome to this keynote about vaccines. My name is Magnus Gisslén. I am a professor of infectious diseases from Sahlgrenska Academy, University of Gothenburg, and we have a very acknowledged sort of speaker. We do have Professor Mark Mulligan from New York University with us, and he is a person that I worked a lot with vaccines. So it will be very, very good to hear your take on this. As an expert, and we will start with this first presentation, and after that we will have a discussion, and then we have a national expert, the Matti Sellberg from Karolinska Institute. Welcome here, Matti. Good to see you too, and most people I think here know you, who you are. First, if you want to put questions, you can see up in the right there is a hand with ask on it. And just put questions there, and then I can take them. We take them during the discussion afterwards, where both Mark and Matti can help answering the questions. So with that, first, it's my great pleasure to welcome Mark Mulligan from New York to us here today, and I leave the floor to you. And just first, a quick question: how the COVID situation is in New York right now?
Yes, well, good afternoon, Magnus and Maktik, and all of those looking in. Pleasure to be here. I regard this as a privilege. We are fortunate in ways right now in New York. You know, March and April, we were truly overrun with COVID. And then things calmed down over our summer here, but they've been slowly creeping up. But just to give you a sense of it, at the worst of it in March and April, in our 3 university hospitals in Midtown Manhattan, Brooklyn, and Long Island for NYU, we had over 800 people in the hospital, and almost half of them in the ICU with COVID. And right now, our total number is roughly 130. So, you know, that gives you a sense. We're not even 20% of where we were, thank goodness, and only about 30 as opposed to over 300 in our ICUs. But we are concerned. The mayor has just shut down indoor dining again. Can still do outdoor dining, but we're actually expecting 12 inches of snow today, which probably isn't much for you, but it's a lot for New York. So not many people will be dining outdoors today.
Well, right now there's no snow in Stockholm anyway. I don't think in Gothenburg either. So you probably have much more snow than we have.
Okay. Magnus, anything else before we start?
Oh, good. So thanks a lot. Please start.
Okay, great.
Well, presentation. Perfect.
Thank you so much. Let me share my screen. And as soon as I have it, please confirm, Magnus, that you can see it. Hold on, sorry.
So
all right.
Yep.
Okay, wonderful. Let's get started. So I'll just draw your attention. Let me get my laser printer going, laser pointer going. I want to just draw your attention to this. Last month, November was extraordinary. Every Monday in November, we had big vaccine news. First, we heard about Moderna's, over 90% protection. Then we heard about Pfizer's 95% protection. Then we had the final data for both Pfizer and Moderna RNA vaccines at about 95% protection. We also had AstraZeneca's news about an average across a couple of studies of 70% protection. So it really was an extraordinary month for vaccine news, and obviously very, very happy, welcome news, giving people hope. But I think for all of us as infectious disease specialists and professionals in this area, public health people need to manage expectations. And what I like to say is that it's not going to be this kind of a thing like an on-off. All of a sudden now we have vaccines rolling out and immediately life is going back to normal. No, it's going to be more like this kind of light switch. I assume you have these in Sweden as well. The dimmer switch that you slowly dial up and the lights come on 10, 20, 30, 40%. And hopefully by mid-summer, we can get maybe 75% of people immunoprotected or vaccinated and achieve herd immunity. And then our hope is to end the pandemic in 2021. So that's that's the image I'd like you to keep in terms of managing expectations. And when you're talking with your patients, with your leadership, your administrators, it's going to be limited by vaccine availability, slowly ramping up. And then I probably don't need to say this for this audience, but I always love to mention that 20 years ago, our US CDC agency awarded vaccines the greatest public health achievement of the prior century, the 20th century. And there it is at the top of the list, along with many other life-saving advances of the last century. But during that century, in part due to vaccines, but admittedly to all of these, life expectancy went from 47 years to 77 years in the US. And now, out of our US birth cohort, which is about 4 million babies a year, there are 40,000 babies and children that grow up that would have died due to childhood infections. So it's really remarkable what the advances we've made, and the number one reason, vaccines. And yet we have work to do in terms of education and trust. And we have vaccine hesitancy at a baseline level, and then concerns about the current situation. Was there government meddling? Was there politicization, politics driving the show? Was safety adequately evaluated? How could you do this with warp speed and have it be adequately safe? So I want to talk about those things. And the reason it's so important is that, you know, now it looks like we have a couple of amazing vaccines in terms of their protective efficacy, but vaccines in and of themselves don't save lives, as my friend Walt Orenstein says. Rather, vaccinations do. So we've got to engage the public, engage our healthcare providers that will be asking to take the vaccine, to be sure they're educated and have all the information they need to make their best decision. And of course, it's their decision. We respect that. The vaccine's not being mandated in the US. It doesn't have full licensure. It has an emergency approval. That's different than an FDA. It has emergency authorization, different than a full FDA approval. So no one's mandating, or at least my institution's not, and CDC hasn't recommended mandating the vaccine for anyone. So we've got to engage and educate the public. And I think we even have work to do with healthcare providers, particularly staff, that may be harder, and minority communities who may have distrust for many good reasons based on the past and ongoing inequities. This point was really underscored when we had the measles outbreak in New York in 2019. And in the US, we had some 1,200 cases that year. We didn't have a child die, but we were very lucky, since we know measles can kill about 1 in 1,000. But we really have to educate. This is me over in Brooklyn, New York City, in an area where there's a heavy concentration of Orthodox individuals, and we're educating them about vaccines. And really, most people are not in the far end of in terms of vaccine hesitancy. They're not rabid anti-vaccine. Rather, they just want to do the right thing and learn and make sure their families are safe. And they want to be educated. They want the facts. So when credible community educators go out, it can make a difference. And this is an example of a flyer or poster for our current vaccine trials. We are currently enrolling in the AstraZeneca trial. We have participated in the Pfizer trial. In fact, on May 4th, the first human beings on the planet to get a Pfizer RNA vaccine were vaccinated at our site and one other site. So we've been involved in it for over 7 months, from the very beginning, the first day that Pfizer vaccines were rolled out. And you can see, you know, this is a poster specifically for our large city hospital, Bellevue, in Midtown Manhattan. And we try to show the types of diversity, the communities that receive their care at this hospital in all the promotional materials. So very important for the educators, the staff at the clinic, to be from the community and look like the community in terms of gaining trust and engagement. Well, let's talk about the candidate vaccines. And I love this quote at the top from Albert Sabin. A scientist who is also a human being cannot rest, while knowledge which might be used to reduce suffering rests on the shelf. Dr. Sabin and also Dr. Salk were both medical students right here at New York University. And we're very proud of that and have a long history of vaccine research here for many other vaccines. But the vaccines that we're going to talk about today are a bit different. They're a new type of vaccine. Well, first, let's talk about the antigen that's being expressed in all of the vaccines I'll talk about today, and it's the spike protein. This exists, as you know well, as a trimer on the surface of the SARS-CoV-2. It is the target of antibody, but also cellular immunity. It can be delivered as a protein, as an RNA, as a DNA, and we'll talk about all of those. The red area here is the receptor binding domain, which is about 200 amino acids in size. This dark blue is one of the trimers, so there are 3 of these in the spike protein complex on the surface of the virion. The entire spike protein is about 1,200 amino acids, so that this receptor binding domain is just a small piece within it. And many of the candidate vaccines are stabilized with 2 proline mutations to stabilize this in a pre-fusion configuration, so that the immune system sees the antigen or immunogen in a conformation that is most important for antibody neutralization. And this receptor binding domain is thought to be a major target of neutralization. Many of the neutralizing antibodies that now have emergency use authorization in the US from Lilly, from Regeneron, target this receptor binding domain. We have monoclonal antibodies with EUA in the US for treatment of individuals as outpatients for mild disease before they require hospitalization and oxygen. And we know that they can reduce the need for hospitalization in people with risk, obesity, chronic conditions, by up to 80%. This displays multiple candidate platforms for COVID-19 vaccines. There are well over 200 candidates in various stages of development, including preclinical, probably 30 or 40 clinical trials ongoing, and probably 10 or a dozen reaching efficacy phase 3 trials. I'd like to talk about one group of platforms, which I'll call the genetic immunization platforms. This one, this one, this one. These all present either an RNA, a DNA plasmid, or DNA within a viral vector, such as a recombinant non-replicating adenovirus. So you're delivering a genetic message to the cell. And if it's DNA, that you make, basically you inject the vaccine into the deltoid muscle of the arm. These are taken up into muscle cells mostly. And you get, in the case of the DNA vectors, you get RNA made in the cytoplasm, and then you make protein from the RNA, the spike protein shown there, and you make the immune response. Same situation with the plasmid DNA. The RNA vaccines, maybe one reason they're so potent and why they've been so effective, they skip that first step because you're not giving the DNA. You go right from RNA into the cytoplasm, never go into the nucleus. There's no integration. This RNA is degraded naturally within days. RNA, messenger RNA is a natural product in the body. So you deliver the RNA and you make the protein, and then you make the immune response. So genetic immunization, these 3 platforms, messaging, in essence, messaging the cells of the body to make the vaccine protein, vaccine spike protein. Perhaps a more natural way to immunize. You get a balanced immune response, both with CD8 and CD4 cells, as well as, of course, B cells and antibody responses. And we'll talk a bit about the innate responses as well. Another platform that I'll mention are the protein vaccines. So here you're not delivering a DNA or an RNA message like you do with the genetic immunization. Here you're just delivering the protein. It's like the standard flu vaccine. In both cases, with the lead manufacturers, Novavax and Sanofi, these are delivered with an adjuvant, the GSK adjuvant partnering with Sanofi. In this case, we'll induce, of course, memory T cells, T helper cells, as well as B cells and antibody, but we're not likely to get a CD8 response with this platform. And of course, there are the more standard platforms that have been used for many years, like inactivated virus, live attenuated virus, and other replicating viral vectors. These viral vectors, the adenovirus of AstraZeneca, for example, is not able to multiply in mammalian cells, in human cells. And so it's a non-replicating viral vector. In the case of AstraZeneca, it's a chimpanzee adenovirus, a serotype that we don't have pre-existing immunity to, or not much. Johnson & Johnson, for their platform, is using an Ad26 non-replicating viral vector. All of these are AstraZeneca, Johnson & Johnson, Pfizer, Moderna are in phase 3 trials now. And all of them except AstraZeneca are fully enrolled. These will be entering phase 3 trials probably in the next quarter or so. We can talk about those more if we have time. So let's go ahead and go to the first of these platforms. I want to talk about this lipid nanoparticle nucleoside modified RNA vaccine of Pfizer-BioNTech. So major pharma partnering with a German biotech company that had worked for years with RNA vaccines in the cancer field mainly. And the RNA is encapsulated in this lipid particle, if you will, 4 different kinds of lipid. It's about the size of a virus particle, gets taken up into cells after injection in deltoid muscle. Pfizer-BioNTech estimates they can produce 50 million doses by the end of this year. So that's enough to vaccinate 25 million people. It's a 2-dose regimen, and 1.3 billion doses by the end of next year. My government paid 1.95 billion US dollars for 100 million doses. That's $19.50 per dose. Storage is logistically challenging with this particular vaccine. Long term, it requires -70. It's good for up to 6 months. This is Arctic cold, -94 Fahrenheit. Short term, it can be stored in a conventional type freezer, like in any kitchen, that temperature, for up to 5 days. And there's some ability to store it on dry ice with certain requirements for up to 15 days. The phase 3 trial for the Pfizer-BioNTech RNA lipid nanoparticle spike vaccine enrolled 44,000 participants in multiple countries. The vaccine was found to be tolerable, safe, and effective. The efficacy was 95% in the final report of November 18th, and also in the recent FDA filing. That efficacy was based on 170 infections that occurred in the trial. And when the DSMB, the Data Safety and Monitoring Board, cracked the code, if you will, looked at which groups, they found that 162 of the 170 infections were in the placebo group and only 8 in the vaccine group. And when you do the mathematics to determine vaccine efficacy, you end up with 95%. And the 95% confidence intervals around that number range from roughly 92 to roughly 98%. So a highly effective vaccine. And it was equally effective in older adults, in all races and ethnicities, and in those with chronic conditions. For each of the secondary analyses, subgroups, at least 92% point estimate for efficacy. It was effective against both mild and severe disease. For example, in this trial, there were 10 cases of severe disease, and 9 of the 10 were in placebo arm. So 1-to-1 randomization, so equal numbers got vaccine and placebo. Again, the platform is a nucleoside modified messenger RNA. The nucleoside sequence non-coding mutations to reduce interaction with TLR7/8, which detect single-stranded RNA. This reduces the immediate and quick innate response and makes these RNAs more tolerable so that we don't have too much vaccine reactogenicity, the expected vaccine reactions. Again, this RNA encodes the pre-fusion spike protein. It includes 2 stabilizing mutations in the spike sequence. And it's a 2-dose series of 30 micrograms of the RNA, which is a volume of 0.3 mLs each. In the US, we started vaccinating Monday, as you know, or as you probably know. They started last week in the UK. There are about half a dozen other countries that have given some form of approval, whether emergency authorization or more regular approval. And the vaccine is being rolled out across multiple countries now. We started Monday. It's gone very well so far. And our national guidelines are allowing the vaccine, because of the very limited supply, to be used for healthcare providers and for those residing in long-term care facilities such as nursing homes. About 1/3 of the deaths in the US have occurred, or roughly 20% of the deaths have occurred in nursing home residents. They've been very hard hit. Just a slide on the antibody responses. So again, I told you we're using this full spike, not just the receptor binding domain for the phase 3 trial. We did study an RNA that encoded just the receptor binding domain in phase 1, but let's focus on the spike, whether in younger adults, 18 to 55, or in older adults, 65 to 85. 2 weeks, let's see, yes, 2 weeks after the second immunization in the 30 microgram, the dose that went to phase 3 and that we'll be going for the international rollout, neutralizing average neutralizing titer in these phase 1 data of 206 compared to human convalescent sera, individuals that have recovered from COVID-19, where they had a median titer of 94 and 50% neutralization titer against live virus, conducted in a BSL-3 facility. And in younger adults, in that same range, 160, 360. So the older adults do quite well, and both of them have higher titers than the convalescent sera. There was a high correlation of IgG binding antibodies with these neutralizing antibody titers, and these sera were able to neutralize a panel of variant viruses, including the D614G receptor binding domain variant, which has become a dominant variant in the US. And we published the phase 1 results in Nature and in New England Journal of Medicine over the last few months. This nucleoside modified RNA also induces T cell responses. And as I mentioned, because we have endogenous synthesis of the vaccine spike protein in the body, we're getting both CD4 and CD8 responses at very nice levels with this 30 microgram dose that's now being rolled out for international use, whether measured in an ICS assay, flow cytometry, or in an ELISpot assay, where you can see, for example, receptor binding domain targeted CD4 and CD8 T cells present after vaccination, but not present before vaccination. And when you look at the cytokines that are produced with restimulation of these memory CD4 and CD8 cells, there's a Th1 polarization. They're producing interferon gamma, IL-2, not IL-4 to a much degree at all. And this is felt to be important because of some theoretical concern about enhancement of disease, what's called vaccine-associated enhanced respiratory disease, an inflammatory response that's associated with Th2 polarization, mainly been induced by protein or killed viral platforms where the formalin had been used to inactivate the virus. So this is good news. This Th1 polarization felt to give us some extra confidence in vaccine safety. And in fact, as I said, we've we've not seen any evidence of disease enhancement. In fact, severe disease is much less common in the vaccine group than in the placebo group. And I'll show you more data on that. Let's change gears and talk about the other RNA lipid nanoparticle vaccine that encodes the spike protein. Again, this one has the two stabilizing prolines inserted to keep it in the prefusion configuration. Moderna says they will produce 20 million doses by the end of this year, so that's enough for 10 million people, and somewhere between 0.5 and 1 billion doses by the end of next year. Storage here is a bit more feasible logistically. A standard minus 20 Celsius freezer can be used for long term, and short term can be stored in a refrigerator for up to 30 days. So this makes it much easier for many outlying clinics where they may not have the freezers. Again, here looking at neutralizing antibody assays, just to show that both younger adults, 56 to 70, and older adults, 71 and over, produce a level of neutralizing antibodies, in this case, an inhibitory dose 80, titers that are very good. And again, although we're not showing it in this case, equal to or exceed convalescent, the titers in convalescent sera. And this paper came out in the New England Journal maybe 10 days ago, just showing now the furthest out immune response data. We've got 3 months after the second dose, and you can see good persistence of binding antibodies in older adults, sort of middle older adults and younger adults. Whether we're looking at binding antibodies or 3 different neutralization assays, the titers are holding steady. There's some drift down. Each of these lines is an individual, so there are about 10 people in each group here. These are phase 1 data, but it's the furthest out immune response data that we've seen for the RNA vaccines, suggesting 3 months out at least from the second dose that we're seeing some drift down, but really pretty good persistence of antibody. And of course, we fully expect that there'll be memory T and B cells there as well. So a point to make, both Pfizer and Moderna together will have enough vaccine to vaccinate only 35 million people by the end of 2020. And I'm talking not just about US, I'm talking globally, the supply. So it's quite limited. The US population is 330 million or 0.33 billion, and that's just 4% of the global population of 7.8 billion. So there'll be a really quite limited supply of vaccine for the next few months. And this is why it's so important. The RNA platforms will not be enough to vaccinate entire countries or to have enough vaccine for everyone who might want it globally. And we do need to think globally. As long as there's disease anywhere, there's risk of disease in Sweden or in the US because of global travel. So we have to think of the global pandemic response. And so we need additional candidate vaccines. We need multiple winning vaccines. And this one, AstraZeneca, is very important. Again, a non-replicating adenovirus vector, expresses the spike protein, and its interim efficacy was reported recently. That came out last week in Lancet publication, but initially was reported a bit earlier. 70% overall, 90% in the UK study, 62% in the Brazil study, and they combined data to come up with this 70%. There were no severe infections in the vaccine group, so again, good protection against severe disease. And they also hinted at least in their press release, some reduction in asymptomatic infection. And that's important because it might reduce transmission. But we need to learn more about this, and we don't have data about prevention of asymptomatic infection for the RNA vaccines, Pfizer and BioNTech yet. They will have that data, it's just not available yet. AstraZeneca will be applying for authorizations in various countries soon, presumably. They have said they can produce 3 billion doses by the end of 2021. So a platform that can make a lot of vaccine. And look at the cost. I told you the US paid about $20 a dose to Pfizer. This is $3 to $4 a dose, as I understand. And look at the storage. It's stored in a standard refrigerator for up to 6 months. So if you think about that, the production capacity of 3 billion, the low cost and the easier storage, this starts to sound like a globally useful vaccine. And this is just one figure from some of their earlier publications, just showing that both, regardless of age group, 18 to 55 or over 70, with 2 doses, you get a titer of neutralizing antibodies that again is equal to or exceeds that of convalescent sera. If you give just 1 dose, you can see you don't get the same level. So the second dose really, for both the RNA and the AstraZeneca, seems to boost the level of antibody. And we know that what's interesting, I'm going to backtrack for a second to the Pfizer vaccine. In the materials that were made public last week for the first time for the FDA expert panel review, a single dose of the Pfizer vaccine in the first 21 days provided 52% protection in those first 21 days before a second dose was given. So there's a relatively small number of infections occurring in that interim period, but it's I think important to acknowledge that it suggests that these are really potent vaccines. But we want everyone to get 2 doses so we can get up to that 95% level of protection. And similarly here with AstraZeneca, the second dose does give you a higher immune response and presumably higher levels of protection. As I mentioned at the beginning, this trial we are currently enrolling. We are an NIH-funded site for studying multiple COVID-19 vaccines. We'll be doing the Sanofi vaccine trial in the new year once we finish enrolling in AstraZeneca. This trial is probably now 3/4 of the way enrolled. It's a 30,000 person trial internationally, and we'll probably be fully enrolled by the turn of the new year. We're very happy to be participating in this important global trial. We have 5 clinics that are NYU NIH-funded clinics across the New York City area. And again, in these phase 3 trials, we enroll those that have higher risk of disease or acquisition of disease. The endpoints for all of the phase 3 trials are laboratory confirmed COVID-19 disease, primary endpoint. All right, well, what is this emergency use authorization? So emergency use authorization is not the same as full approval. It's allowing us to use the product in the emergency, and it's a decision by FDA that the known benefits outweigh the known and potential risks. And we now have this emergency approval for Pfizer, and we're rolling out vaccine this week in the US in all 50 states. And Moderna has its advisory review tomorrow. Its advisory committee review, and we believe FDA will provide an EUA emergency approval for Moderna probably either tomorrow or the next day, Friday. Well, how do we allocate? Demand is going to exceed supply in the first several months of any national program, and so decisions have to be made when there's an imbalance between supply and demand. This is a national hero in the US, Dr. Fauci, as depicted by a street artist here in Lower Manhattan. I was on a bike ride a few days ago and came across this street art, which I really liked. So this is meant to depict limited supply in this early phase, and eventually we'll get maybe by mid-year 2021 to have enough doses for anybody that wants to get the vaccine. But initially it's going to be limited, and so decisions have to be made. The principles that lie behind this are to try to decrease death and disease as much as possible, preserve the functioning of society, reduce the extra burden the disease is having on those already facing disparities, and increase the chance for everyone to enjoy health and well-being. So CDC has used these guiding principles to help them in making their recommendations. And there's a fairly large group that they'd like to have the vaccine be rolled out to before it goes to, say, your average healthy 30 or 40-year-old. And they're shown here, and the approximate sizes of these in the US. So healthcare personnel, we have about 21 million. Nursing home residents and staff, about 4 million. These are the first two that in the ACIP has now recommended in a publication that came out just 10 days ago, roughly. Healthcare personnel and long-term care facilities will be offered the vaccine first, and that is indeed who it's being given to in the US this week. And the rationale, again, we've had nearly 1,000 healthcare provider deaths and 70,000 deaths in nursing homes, as I've said. About a quarter of 20% or a quarter of US deaths. And this has been the timeline. CDC issued its prioritization December 1st. On December 10th, the FDA Advisory Committee met. It'll be meeting tomorrow for Moderna. It was for Pfizer on the 10th. Approval granted on the 11th. We think Moderna will get it this week in the US for emergency use. And the vaccine was distributed over the weekend, and we started vaccinating on Monday in the US. And again, in the US, I apologize for being a bit US-centric with this slide, but this is what I know best. 22 million people will be able to be vaccinated by the end of the year. It's only 7% of our population. So we're only going to have, with both Pfizer and Moderna, about 22 million people be able to be vaccinated, 2 doses. And of course, our goal, as I said at the beginning, is to achieve herd immunity. Some people say we should call this community immunity, since we live in communities, not herds. But in any event, it's been estimated that perhaps with an R0 of 4, the average number of secondary cases from a primary case being about 4. Now that you average that, given we know there are super spreaders, but in any event, when the population is fully naive, as we essentially are to this virus, this novel coronavirus, the 4 seasonal coronaviruses don't seem to be providing much cross protection. Everyone is susceptible and can become infected, on average 4 per primary case. But then if we can get people immune with vaccines or perhaps from natural infection, we can shut down the pandemic once we achieve a 1 - 1 over R0, or about 75% of the population vaccinated. When you get the R0 down to less than 1, the pandemic will die out over time. And that's our goal. And we have to protect the vulnerable members of society, those that may not be able to respond well to vaccines, too young, too old, or immunocompromised. So it's inherent on us, even if we don't think we need the vaccine for ourselves, to be altruistic for those around us who might have a fatal infection. And so again, back to as I move to close here, back to the power of vaccines. Smallpox now eradicated. And this was the New York City Smallpox Hospital on an island in the East River. It's now a decaying, crumbling ruin thanks to the vaccine. And I want to thank our research clinics, our participants, our administrators and funders, and just my last slide say that as we advance towards a vaccine for COVID-19, and we're now rolling it out, we've had good progress, but we need more candidates to fill the need. We don't know the duration of protection, or if we'll need a late booster. We don't know the immune correlate of protection. Is there a certain titer of antibody that is a threshold of protection? What about long-term safety? What about prevention of asymptomatic infection? Real challenges include vaccine uptake. I talked about gaining trust, rebuilding faith in science, in clinical trials, the logistics, the freezers, the limited supply, and having to prioritize. And my request to all of you as leaders in public health, infectious diseases, medicine, is to be a vaccine ambassador, to learn about the vaccines and help with this need to gain faith and trust and educate. And as I said at the beginning, vaccines don't save lives, vaccinations do. So with that, Magnus, I thank you and the organizers. It's been my great privilege to share this information.
Thank you so much for this very clear and sort of comprehensive review of the field. We have had a lot of questions actually from the audience, but before I go through some of those, I will see if Matti has some comments or thoughts about this. So Matti, what do you say?
Yeah, I think I really like to thank you for a really exhaustive and thorough and fantastic review of the field. I mean, really great. Really appreciated it. And I think your message is that as experts, we really have to be ambassadors, because what you hear every single day is the pandemics. You hear about autism, and I know that UK exported Andy Wakefield to the US, unfortunately. So it's, I mean, it's really important that people have a trust in the vaccines. But just some quick, I think something that the field as a whole has kind of shown now. It's so nice when you can follow a story from the mouse that was vaccinated or immunized until a protected human. Then you can really get a feel for what does these preclinical data say? What does it mean? And I think if you compare, for example, Moderna and Astra, the Astra vaccine had probably tenfold lower antibody levels in the mouse. It showed slightly less protection in the macaque model than Moderna, and the same thing in humans. So it really follows a clear pattern. I mean, do you have any comments on that?
Yeah, that is interesting, and I have sort of tracked those same observations myself, Matti. I think you're correct on that, and you know, we were thrilled with 95%. It sets a high bar.
Oh yeah.
maybe not to be achieved by the other platforms, but make no mistake, Dr. Fauci came on our team call the other day for the AstraZeneca sites, and he said, listen, don't think this isn't important. We need all winning vaccines out there.
Yes.
As he has said many times, he would have been thrilled if the RNAs had been 70% protected. Yeah. The fact that they were 95 was just amazing. So I think there's plenty of room. In the US, the FDA has said it'll license a vaccine that's safe, that has good manufacturing, if it's at least 50% protected. Yeah. We would love it if the other vaccines come in, you know, in the 70% range or thereabout. So that's, you know, that's my thought. And I do agree. It's many times vaccines have taken so many decades to develop that no single human gets to see that whole tracking that you just talked about, from preclinical to phase 1, 2, and 3. We've been able to observe that in 10, 11 months. It's truly extraordinary.
Yeah, it's amazing. And then just, so you're now participating in the Astra phase 3. What dose regimen? I'm curious. Are they doing with the low high dose or the high high?
It's so this trial was designed and launched and well underway before that information came out about a year ago. So we're high, and this trial will be completed. And you know, the dosing interval is fixed at 28 days. So it's a standard approach. There, you know, there is some biological plausibility to this low high thing, and I think it should be investigated. The thought being, the prime may induce vector specific immunity, and so when you come in with a lower prime, there's enough there immunologically to prime against the insert, the spike, but not so much that you overwhelm with the boost the ability to get a good take. So maybe there's a better take with the low high, but in any event, there were so many other variables with the interval. It's hard to get a good interpretation on that. It should be studied, you know, when you can in the future. Yeah. But our study will go ahead, very high quality, you know, NIH type trial, and we'll see what we.
There has also been some discussion about the possibility of acquired immunity against the vector when you have a high dose. Do you think there is any data supporting that, or is it difficult?
Yeah, no, I do think there is data to support that for years with other adenovirus vectors. We've used them for decades, a couple decades, for HIV and many other things. And some companies, like, for example, the Russians with their adeno platform, they're using two different serotypes of adenovirus to try to get around that issue of having a first dose induced vector specific immunity that dampens the take of the boost. So yes, there is evidence for that with other, with this one, you know, I'm not sure I've seen evidence for the current AstraZeneca, but you know, they didn't get much of a boost with their T-cell response from the data I saw in the phase 1 trial. You do get some boost of their antibody response.
Another question here is, what about, how do you see, and I ask both of you, for vaccination of people that already have had a COVID-19 infection? How do you do in New York, and what do you say, Martin?
We are doing that. CDC has said it's fine, and the clinical trial included many people. And I mean, we didn't screen for antibody to rule people out, exclude them. If they had a known COVID infection, they were excluded based on that. But many people, CDC estimates 40% have asymptomatic infections. Also in New York, in the early weeks and months, testing was impossible to get. So there were many people enrolled that had antibodies, had prior COVID infection, and they do get a boost, and they did get protection. And there were no safety concerns. So based on that, although the numbers are relatively small, the US authorities are taking a permissive approach to it. And I actually feel that's the correct approach. It may be true that in the first few months after natural infection, there'll be protection, but we know that you can be reinfected probably after several months. So I think when we're trying to end the pandemic, it makes some sense. You could make an argument that maybe don't vaccinate in the first 3 months to help with the supply issue, but that gets more complicated. How do you do that?
I completely agree with you that the major issue, should we pre-screen everybody? I mean, that's going to be a logistical nightmare. Yeah. And as you say, the data suggests there's no antibody enhancement in disease. There is no such danger signals. So I think, I mean, probably one would advise people, if you know you've had it recently, you wouldn't need to don't stand first in line. Yeah. But I mean, we're going to vaccinate the risk groups, hospital staff, and the caretakers first. So, but one follow-up question on that: how long would you guess that the immunity from the vaccines persists? Yeah. I mean, I would say, if I would guess, at least 6 months wouldn't be a bad guess. Maybe up to a year. What's your guess?
My guess is the same. But I want to underscore guess, of course. But, you know, just looking at, we always see antibody drifting down. Those Moderna data look pretty good to me 3 months out. And it's such a high level of protection. Even if it drifts down, you know, maybe there'll be some diminution in protection, but it's still going to be very high, I think, even between 6 and 12 months.
We are soon sort of have to round up, but first I will ask a question about sort of beyond the COVID when it comes to RNA vaccines. I suppose that this is just a start also for other sort of infections and maybe. So what do you think of this? Will this be the start, and in such case, which will be the next RNA vaccine that will be? Yeah.
I think it's a natural way to let your mind wander of, you know, the many needed vaccines that we've been struggling to develop, if this could make a difference. So I think that will be explored, you know, maybe HIV, for example. But I also think most importantly is that this was actually planned as and executed beautifully as a platform for rapid responsiveness, for pandemic preparedness. We now know that you can, in months, you know, test in humans and determine efficacy with a pre-existing platform, where next time, whatever it is, let's say pandemic flu, instead of putting in the coronavirus spike, we'll just put in the flu HA very, very quickly, and then you can be out and going. So because it's a rapid response technology, I think we're going to see it used with every future pandemic.
It's a fantastic development.
Just a final comment. I got a mail here, and I think that's important. Do you think, or do we think that the vaccine will help us reach herd immunity? And my answer is, without the vaccine, we probably will never reach herd immunity unless we let the virus run free, and nobody wants that. So I think the only way to get herd immunity is through the vaccine.
Absolutely, I agree. I think relying on natural infection to achieve herd immunity would be devastating in terms of deaths. We're having 3,000 people a day die in our country right now. It's staggering. So we believe in the vaccine and that we can and will achieve herd immunity. Probably 75% or so need to be vaccinated, and that'll be tough. We've got a lot of work to do, but it's good. That's why we're here.
I think that was very good word to end up this session. Thanks a lot, Mark Mulligan and Matti Sellberg. This was excellent. Thanks a lot. Bye-bye.
Thank you, Magnus. Thank you.